Good evening, ladies and welcome you to this class of parastory. And from your occupational standard, it is referred to as apply parastoological techniques. Parastology refers to the study of parasites, and parasites are mainly organisms which live in or on another organism, and they derive benefits such as protection, shelter, and food, but they adversely affect the animals which host them, but they don't destroy rapidly. And so these parasites consist of a number of organisms, basically the unicellular protozoa, and we have the multicellular parasites, which are divided into flatworms, roundworms, and we also have the arthropod vectors. And in this lesson, we are going to start with protozoa, and the study of protozoa is known as protozoology. And so before we go into the finer details of protozoology, we need to understand a number of terminologies which are used in parasitological studies. The first one being parasitism. Parasitism refers to a biological association of two individuals or populations of different species in which one parasite benefits at the expense of the other, which is the host, and which it often affects adversely but does not rapidly destroy. Essentially, parasites are supposed to derive nutrition, shelter, and protection from their host, but eventually in the process of deriving that advantage or benefit, the host suffers, and gradually it might even lead to death or immunization or debilitation. Depending on the load and the number and the type, parasites can cause more or less harm to the host, and also the host, depending on its size and immunity status, it can also be able to resist the parasite. But however, in one way or another, the parasites affect the hosts adversely, though gradually. The next terminology is ectoparasite. An ectoparasite is a parasite that lives on the surface of its host. Many of the times we see takes on livestock, lice on our bodies, mites on chicken and other livestock. These are some of the ectoparasites, and they live specifically on the skin of an organism, whereby they suck blood and cause damage of skin and loss of blood through the pores they make through the skin as they feed, alongside transmitting various diseases. Then we have the next group of parasites known as the endoparasites. These are the parasites that live inside the body of their hosts. We all know about the worms, the various worms, the tapeworms, which live in the intestines, some live in the lungs. We also have other parasites which live within the intestines, some within the urogenital system or the genitalia. These all form a group of parasites known as endoparasites. Then we have another terminology, parastasis. Parastasis refer to the condition where a host carries a parasite or parasites within itself, but it does not show outward signs of having the parasites or of suffering or of disease. Then we have parastosis. This is a condition where an organism has the parasites and shows outward signs of the disease or infection. Then we have parasthenia. This one refers to the amount of load or load of parasites in the blood of the host. And this is essentially an indicator of the degree of an active parasitic infection. Besides that, we have facultative parasite. This is a type of parasite that is capable of living as a parasite, or it can live freely. That means it can exhibit both parasitic and non-parasitic modes of living. And the two very most important parasites that show this behavior are the Nigeria fowleri and Strongyloides stacularis, and these are protozoan and nematelmints, which we are going to meet as we proceed with our parasitological studies. We also have another group known as the obligate parasites. These obligate parasites strictly live as parasites, and it can by no means exist without the host. In other words, these organisms strictly and mainly depends on the host for nutrition, shelter, protection, and transportation. Such an parasite is the Plasmodium, which causes malaria, and Trichomonas vaginalis, which causes trichomoniasis, which is a venereal disease caused by protozoa. Then besides that, we have permanent parasites. These are parasites that live for the greater part of their lives in adults. Or rather, they live as adults in their hosts, such as lice, fleas, and tapeworm and coccidia. Then we have accidental parasites or incidental parasites. These are parasites that accidentally gain entry into an unnatural host and it manages to survive. Okay, welcome again. So besides the various terminologies which refer to the types of hosts and the types of parasites, we also have other terminologies which refer to the various morphological forms and various developmental stages. We're just going to look at a few of those, so that we can move to the classification straight away. So we have So trophozoite, these are the active, motile, feeding and reproductive stages of the protozoan life cycle. Then we have the cyst, is a dormant, non-motile stage surrounded by a protective wall or a thick wall that is essentially supposed to make it resistant to environmental conditions that are harsh, such as salinity, desiccation, and pH, and even chemicals, not forgetting high temperatures or heat, or the dry spell. And these are normally found in an environment outside the host. Then a brother to it is the oocyst, which is a reproductive stage, which also has a thick protective cell wall, but basically it is found in the tissues within the host and is a reproductive cell. So existation refers to the process by which the protozoan emerges from its cyst stage to become a trophozoite, while encystation, which is the exact opposite of existation, refers to the transformation of a trophozoite into a resistant cyst. Besides that, we can go to another one known as the pericle. This is a thin, flexible, or rigid outer membrane that gives a certain protozoa the definite shape. We're going to see the types of shapes in a moment. Then we have the cytoplasm, and in these protozoan parasites, it's divided into two distinct layers. We have the outer transparent and relatively rigid layer, which is called the ectoplasm, and the endoplasm, which is the inner granular and more fluid part of the cytoplasm, and this is where all the organelles are embedded. Then we have the cytostom, which is a specialized provision or cell mouth used by some protozoa for ingesting food or for trapping the prey, as we are going to see in the next few subtopics. Then we have the contractile vacuole, which is an organelle used for osmoregulation within the cell, and normally it is used for pumping excess water out of the cell, and in the process it takes part in the excretory process. Then we have the organs of locomotion, which are basically three types in protozoa, and they form a very important aspect in the classification of the protozoa. The first one refers to the pseudopodia, which are temporary cytoplasmic extensions used basically to capture prey and to facilitate locomotion or movement. And these are common in the mastigophora or the amoeba. Then we have the flagella, which are long, whip-like appendages, which are used for propulsion, and we're going to see in a moment where they are found. Then we have cilia, which are short hair-like structures that fit in coordination to facilitate movement and feeding in where they occur. And these two, flagella and cilia, are common locomotor structures, which I hope you conveniently covered when you were doing your cytology and histology. Then we have the undulating membrane. This is a thin sheet-like extension, and it's normally associated with the flagella, and it is important during the movement of the parasites within the body tissues, which could be the blood, plasma, or the cytoplasm. And this is common in trypanosomes, though other protozoan parasites possess this, such as the trichomonas species. Then we have binary fission. Binary fission is an asexual process of reproduction whereby a single cell divides into two equal parts, giving rise to two identical individuals. Then we have conjugation. This is an asexual process which normally occurs in ciliates, which involves temporary exchange of nuclear material between two individuals. Then under the types of hosts, we have the definitive host, which is the host in which the parasite reaches maturity and undergoes sexual reproduction. We also have the intermediate or secondary host, whereby the asexual and immature stages of the parasite are harbored or are hosted. We also have an important host known as the reservoir, which acts as a source of spreading of the parasite. And this host normally does not show any signs of sickness, but it has the parasites within and is capable of transmitting it to other hosts. And this brings us to the end of the terminologies that are used in parasitology, or rather in protozoology and parasitology in general. Now, under classification of the protozoa, it would be wise if we started with the general classification of parasites. And so parasites are broadly classified into the unicellular and the multicellular eukaryotes. And under protozoans, we have 4 classes. And under metazoa or under multicellular parasites, we have a number of flatworms, which includes the flukes and the tapeworms. Then we have the roundworms or the nematohelminths or the nematodes, and here we have quite a number, and we are going to discuss about them. Then we also have parasites of the arthropods, and these majorly acts as vectors, although they may directly act as parasites. And this includes the ticks, mites, and insects, which are important disease vectors or per se parasites. The protozoa, since time immemorial, have been classified into 4 major groups based on their locomotor structures or primary mode of locomotion or movement. But however, currently we have molecular classifications, but classification according to the locomotory structures still remain in force and is being used up to date. So the first category of protozoa are known as flagellates, and they fall under the Mastigophora, and these simply move using one or more whip-like structures known as the flagella. And these parasites include the free-living forms and the parasitic forms. The free-living forms include the euglena, while the parasitic forms include the trypanosoma, leishmania, trichomonas, and giardia species. The other group move and feed using temporary cytoplasmic extensions called pseudopodia, which is a terminology that means false feet. And the two common examples that are under this sarcodina or the amoeba include the amoeba proteus and the entamoeba histolytica. We are going to discuss about this in a moment. Then the other class of protozoa or category of protozoa include the ciliates, which belong to the Ceriophora, and they move with the help of short hair-like projections that beat in coordination. And these groups consist of free-living and parasitic forms, whereby the free-living forms are represented by the paramecium, while the parasitic groups or the parasitic ciliates are represented by the Balantidium coli. The rest of the protozoans that are remaining do not possess any organs of locomotion, but they are simply non-motile in the adult stages, and they are exclusively parasites, and they are obligate intracellular parasites. These have complex life cycles and often use very special apparatus known as the apical complex to invade the host cells, hence the name apicomplexer. And examples include the Plasmodium, which causes malaria, and Toxoplasma species, which causes toxoplasmosis. Now, with that brief introduction, then let us look at individual key characteristics or salient features of each class of protozoa, and we'll start with the sarcodina or the amoeba. Very important to start with is the mode of locomotion, is that they move with the help of the false feet or the pseudopodia. Then this class has existing the trovozoite and cyst forms. They multiply by binary fission, and most of them inhabit the intestinal tract, or simply the intestines. And among the common parasites of medical importance, we have Entamoeba histolytica, which causes amoebic dysentery. Then we have Entamoeba coli, which is actually non-pathogenic, but an indicator of fecal contamination, just like its counterpart, Escherichia coli, which is a bacteria which is used as an indicator for fecal contamination of food and water. Then we have the mast gopher. These are the flagellates, and they move with the help of the flagella. And they may have one or multiple flagella which aid in motion. And with me here are a number of those species, and I've selected very clear diagrams, which or rather photos, which have been taken after staining has been done. So in the first picture or template, we have the Trypanosoma species, which is an obligate intracellular parasite. We can see the part that is thin and looks like a tail. That's the free flagellum. But on top of it, we have the other flagella, which runs along the margin of the undulating membrane. Then that flap you are seeing actually is the undulating membrane, and this is supposed to assist the trypanosoma in the movement within the fluid. Remember, this is blood, and these are red blood cells, while the trypanosoma is moving through the plasma or blood fluid. Then when you look at the next one is the Trichomonas vaginalis, which causes trichomoniasis, which is a venereal disease. This one has a number of flagella. We have the anterior flagella, which are 4 in number. And then we have the Gaillardia duodenalis, which has a set of 8 flagella, whereby we have 2 the anterior flagella, we have 2 caudal, then we have the 2 posterior, and the 2 ventral flagella. And in total, we have 8 or 4 pairs of flagella. Then when we look at the fourth one, this one is a plate that contains the leishmania, which causes a skin infection, which we'll study later. It has a single flagella, and so these mastgophora or flagellates rely on these flagella for their locomotion, and they have some which are intracellular, or those which live within the tissue. They have the undulating membrane to assist or to aid the organism or the parasite in swimming through the fluid. Remember, this trichomonas is moving through the vaginal fluid, and it's a common problem or a serious problem with the females. It also has an undulating membrane to help in movement through the fluid medium within the genitalia. And so depending on the preferred site, the mastigophora or the flagellates are divided into a number of subgroups, which include the intestinal and urogenital flagellates. These are tissue and blood-dwelling parasites. And within this urogenital and intestinal, we have Giardia ramblia and Trichomonas vaginalis. Then blood and tissue parasites include the Trypanosoma species, which causes African sleeping sickness and Chagas disease. This is American trypanosomiasis. And then we have the Leishmania donovani, which causes leishmaniasis. Then the Cereophora or the ciliates, which are represented by the Balantidium coli, as we have discussed shortly before. We said they move with the help of cilia, which are hair-like extensions on the cell surface, which they beat in coordination to assist in the feeding and motion. We can see that they have it has the cytostome, which is the cell mouth for feeding, and the area, or rather the hair-like projection surrounding the cell. And the most important representative species for this is the Balantidium coli. Then we have the sporozoa, which we said are non-motile, and they have special apparatus for invading the cells, hence the apicomplexan. Then they have complex life cycles, which constitutes the asexual and sexual stages. And one other unique property or feature with them is that they are strictly obligate intracellular parasites. And these are represented by the Plasmodium, which transmits malaria or causes malaria. We have Toxoplasma gondii, which causes toxoplasmosis. We have Cryptosporidium and Isospora belli. These are typical intestinal coccidia, and they have no defined organ for locomotion, but they depend on the host for survival and movement. And this summary, or this synopsis gives the very key characteristics for each group, and they differentiate each group from the other. And at this juncture, I want us to now look at the very general features that put an organism into the protozoa, and they include: one, the general morphological forms, that is the cell structure, the cell size and shape, and then the major morphological forms. So for the general cell structure, a protozoan parasite will always be unicellular, or it will be a single cell, and they have a true nucleus, hence the name eukaryotes. And this cell will have an outer covering known as the plasma membrane. This plasma membrane allows flexibility, allowing change of the shape. They also, within the plasma membrane, they have cytoplasm, and this cytoplasm is unique in that it is divided into two distinct layers, which forms the ectoplasm, which is the outer and the clear layer, while the endoplasm is the inner layer, and that is granular. Then besides that, they may have one or more nuclei or multiple nuclei, we shall be discussing as we go to the individual species. Then, as we need energy, every cell also requires energy, and the energy is normally synthesized in special organelles, which we call the mitochondria, and each cell must have mitochondria to serve the energy requirements for the cell as it carries out the various metabolic activities. Then most of the protozoa, as much as they are unicellular, they normally have specific shapes, with the amoeba being variable because it keeps on changing shape as it develops the pseudopodia for purposes of movement and feeding. From the diagrams you have seen above, we can see some are pear-shaped, like the Giardia and the Trichomonas, they are pear-shaped. Some, like the Trypanosoma, are leaf-like or spindle-shaped, while the cyst forms, especially the cysts, are spherical or oval in shape. Then for each of the parasites, we have the trophozoite stages, except the Plasmodium, and these are the active feeding and motile stages or mobile stages, and these are normally found in tissues or body fluids. Besides, these are delicate outside the host, and so they are vulnerable, and so don't exist outside the host. And they are the ones which are responsible for the pathological conditions in the hosts in which they reside. Besides the trophozoite stage, we have the cyst or the oocyst stage, and this basically are simply protective stages and reproductive stages during reproduction and protection, as the parasite finds its way into the outside environment where it is supposed to infect more hosts. And these oocysts and cysts, they are dormant, they are non-motile, and they are the ineffective stages. They are the ones that if infect new hosts, they are thick-walled and resistant to the environment, meaning that whenever they are in the environment, they cannot be easily affected by acids, by chemicals, heat, chemicals, or salinity, and they are very critical in the transmission of the parasite from one host to another. And as we have said, they are either round or oval. They have a very thick protective wall, and because they are dormant, then they have greatly reduced metabolic activity, and besides that, they may have one or more nuclei, and the younger the cyst, the lesser the nuclei, and as it matures, most of them contain 4 nuclei, hence quadrinucleate cyst, or some may have 2 nuclei. But basically, they have multiple nuclei once they are mature. Now, having had this summary of a rather lengthy discussion of the morphology of the various classes of protozoan groups, then we want to ask ourselves, why is it important for us to understand the morphology in parasitology and even in protozoology? Very important, and on top of the list, is that the various morphological features form the basis of laboratory diagnosis. It helps us to understand whenever a host has a given disease, and then you take blood or you take sample for the relevant host, then what are you expecting to find in that? We cannot tell unless we relate it to past experience. Hence, once you understand the morphology of one organism from another, you can be able to tell that indeed the animal is suffering from this parasite because of these observations of the parasite in the specimen that has been collected and that has been used for the diagnosis. Besides that, once the habit of a parasite has been known, then we can be able to actually link the pathogenicity to a certain parasite, and by the time we realize that it is the one or not the one in the sample, then we can be able to distinguish between the pathogenic and the non-pathogenic species. Besides that, the morphology guides in the treatment and control of the parasite, and then in the spread of diseases. We know that some diseases are endemic in some areas, while in other places they are very rare. So this morphology is quite very critical in identifying the type of parasite which is endemic in a given place, or it's also important in knowing or in identifying the emerging strains or species, because the ones that are commonly known to be endemic in a certain place, their morphology is known. So these morphological studies are very key in epidemiological studies of such a protozoa. So I think up to this much, we have actually done a good job, and I hope as we delve into each of the species, we'll have easy time to learn more about the other aspects by relating to the various aspects we have discussed, especially about the classification and the morphological features. Thank you, and wish you the very best till we meet again.